What is DNA polymerase? DNA polymerase is an enzyme responsible for synthesizing DNA strands during DNA replication. Hello, I'm Luna, the wise owl scientist. Today, we're diving into new concepts Luna style, making them not only interesting but easily understandable. So, without further ado, let's get started and explore this exciting journey together. In which cellular process does DNA polymerase play a crucial role? DNA polymerase plays a crucial role in DNA replication, where it ensures the accurate and faithful copying of the genetic information. What is the primary function of DNA polymerase in DNA replication? The primary function is to catalyze the addition of nucleotides to the growing DNA strand, following the template provided by the existing DNA strand. How does DNA polymerase ensure accuracy during DNA replication? DNA polymerase has proofreading capabilities, enabling it to correct errors by removing and replacing mismatched nucleotides. Name the three main types of DNA polymerases in humans. DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon are the three main types in humans. What is the role of DNA polymerase in repairing damaged DNA? DNA polymerase is involved in DNA repair mechanisms, helping to fill in gaps and correct damaged or mismatched bases in the DNA sequence. During DNA replication, which DNA polymerase synthesizes the leading strand? DNA polymerase 3 synthesizes the leading strand continuously in the 5'3" direction. Which DNA polymerase is responsible for the synthesis of the lagging strand? DNA polymerase 3 synthesizes short fragments on the lagging strand, while DNA ligase joins these fragments. What is the direction of DNA synthesis catalyzed by DNA polymerase? DNA synthesis occurs in the 5'3" direction, meaning new nucleotides are added to the 3' end of the growing DNA strand. What is the significance of the 3' to 5' exonuclease activity of DNA polymerase? The 3' to 5' exonuclease activity allows DNA polymerase to proofread and correct errors by excising mismatched nucleotides. How does DNA polymerase distinguish between the correct and incorrect nucleotides? DNA polymerase discriminates based on the proper base pairing between the incoming nucleotide and the template strand. Name a syndrome associated with mutations in DNA polymerase genes. Cockayne syndrome is associated with mutations in DNA polymerase genes, leading to impaired DNA repair mechanisms. What is the role of primase in the context of DNA polymerase? Primase synthesizes short RNA primers that provide a starting point for DNA polymerase to initiate the synthesis of a new DNA strand. Why is DNA polymerase 3 considered the primary enzyme in DNA replication? DNA polymerase 3 is the main enzyme responsible for elongating the DNA strand during replication, ensuring efficient and accurate synthesis. Can DNA polymerase initiate the synthesis of a new DNA strand without a primer? No, DNA polymerase requires a primer, usually an RNA primer synthesized by primase, to initiate the synthesis of a new DNA strand. What is the role of DNA ligase in the process of DNA replication? DNA ligase seals the nicks between adjacent DNA fragments on the lagging strand, creating a continuous and fully functional double-stranded DNA molecule. How does DNA polymerase contribute to the fidelity of genetic information? DNA polymerase proofreading and error-correcting mechanisms help maintain the fidelity of genetic information by minimizing replication errors. In eukaryotic cells, where does DNA replication take place? DNA replication occurs in the cell nucleus in eukaryotic cells. What is the role of DNA polymerase I in prokaryotic DNA replication? DNA polymerase I in prokaryotes is involved in removing RNA primers and replacing them with DNA during DNA replication. How does the processivity of DNA polymerase contribute to its efficiency? The processivity of DNA polymerase allows it to remain attached to the template strand, facilitating the synthesis of long DNA strands without frequent detachment. What is the relationship between DNA polymerase and the sliding clamp? The sliding clamp, a protein complex, helps DNA polymerase maintain contact with the template strand, enhancing processivity during DNA replication. How does the fidelity of DNA polymerase affect genetic stability? The high fidelity of DNA polymerase contributes to genetic stability by minimizing errors during DNA replication, ensuring accurate transmission of genetic information. Can DNA polymerase replicate both strands of DNA simultaneously? No, DNA polymerase replicates the leading and lagging strands sequentially during DNA replication. What is the role of the 5' to 3' exonuclease activity of DNA polymerase? The 5' to 3' exonuclease activity of DNA polymerase is involved in removing RNA primers during DNA replication and in proofreading the newly synthesized DNA strand. How does DNA polymerase contribute to the fidelity of genetic inheritance? DNA polymerase ensures accurate replication of genetic information, contributing to the fidelity of genetic inheritance across generations. In what cellular compartment is DNA polymerase synthesized? DNA polymerase is synthesized in the cell nucleus and then transported to the site of DNA replication or repair. What is the role of the template strand in DNA replication? The template strand provides the sequence information for the synthesis of a complementary DNA strand during DNA replication. How does the fidelity of DNA polymerase relate to the prevention of mutations? The fidelity of DNA polymerase reduces the likelihood of introducing mutations during DNA replication, thereby preventing genetic abnormalities. What happens if there are errors in DNA replication that escape correction by DNA polymerase? Unrepaired errors can lead to mutations, potentially causing genetic disorders or contributing to the development of cancer. Can DNA polymerase function in the absence of other accessory proteins? While DNA polymerase can catalyze the synthesis of DNA, it typically requires the assistance of various accessory proteins, such as primase and DNA ligase, for efficient and accurate replication.
Thank you so much for watching. I appreciate your time and support. If you enjoyed this video, please consider subscribing to our channel and ringing the notification bell so you won't miss any of our future content. Your support means the world to us, and we can't wait to share more exciting videos with you.